Okay, welcome to KIN 225. I'll be delivering the rest of your lectures remotely. I'm going to try to do this via YouTube. This is my first attempt to upload a YouTube video for your course. This is my technician that's helping me out navigating YouTube and how to create a YouTube channel. Here is my training partner. And let's get to your lecture here. So a few announcements first, and I've posted this all in your PowerPoint file. So Lab 6 and 7 are due a week later than originally scheduled. So Lab 6 is due Monday, March 30th, Lab 7th, Monday, April 6th, and everybody is to hand in the labs on those days. They're handed in online. I think Vanessa or Doug are going to make arrangements and let you know how to hand those in online through the Blackboard system. The final quiz for the course has been canceled, so the 15% worth of quiz marks for your final grade will be just based on your first two quizzes. The final exam is being scheduled on the original date planned, so starting at 9 a.m. on Saturday, April 25th. It's going to be delivered online, and you're going to have you're essentially going to have an unlimited amount of time to do that exam based on the university's policies. It's going to be 120 multiple choice questions, 30 questions from material before the midterm, 90 questions based on material after the midterm. So majority of questions are from after the midterm. Today's class is going to be a review of glycolysis, so going from anaerobic to aerobic, a question on whole body lactate metabolism, and then the main focus will be on kind of a summary of carbohydrate metabolism going from glycolysis through the Krebs cycle and the electron transport chain. And we're going to do that by identifying all the steps where ATP is actually produced. So first review question here is with anaerobic training, the ratio of lactate dehydrogenase M to your lactate dehydrogenase H would increase. So lactate dehydrogenase M is your muscle form. Lactate dehydrogenase H is the heart form of the enzyme. And the answer to this question is false. It is false because I've got the two enzymes outlined here. The lactate dehydrogenase M form is outside the mitochondria. The lactate dehydrogenase H is inside the mitochondria. And as I mentioned before with aerobic training, your mitochondrial enzymes will increase, but your cytoplasmic enzymes, the enzymes outside the mitochondria, will pretty much stay the same. So if I go to the whiteboard and we're considering the ratio of lactate dehydrogenase M to lactate dehydrogenase H, with aerobic training, you're getting an increase in the mitochondrial enzyme, no change in this enzyme. So the ratio, if anything, would actually go down because this is staying the same and this is increasing. So the correct answer to this question is false. This question has to do with the review of whole body lactate metabolism. So aerobically trained individuals feel less stressed when they're exercising at high intensities. This results in less release of what are called stress hormones, and that includes epinephrine. Epinephrine is just adrenaline. And the epinephrine has one effect one of its effects is to cause constriction of blood vessels. So it makes your blood vessels constrict and impedes blood flow. So the question here is, what is an additional reason that trained individuals have lower lactate levels compared to untrained individuals working at the same exercise intensity? And to answer this question, you have to go back to last day and look at the Cori cycle. So the Cori cycle involves removal of lactate from the blood to the liver where it's converted to glucose. And this occurs during exercise. So with a trained individual, if they have less release of epinephrine, they're going to have less vasoconstriction, they're going to have greater blood flow to the liver, so greater lactate delivery to the liver and removal of lactate through this process. So that's an explanation for why they could have lower lactate levels compared to untrained individuals. Recall another reason is that lactate, once it's produced in the muscle fiber, can go to mitochondria and be oxidized, or it can go into the blood and be taken up by a more aerobically, a muscle fiber with greater aerobic capacity and be oxidized in that mitochondria. So to summarize, an aerobically trained individual will have lower lactate levels than an untrained individual because they get greater lactate uptake by the liver, and also they'll have greater lactate oxidized in their mitochondria, and that leads to lower lactate levels. Another review question here: which of the following is a mitochondrial enzyme that has acetyl-CoA as an end product of its reaction? The correct answer here is B, pyruvate dehydrogenase. So your phosphofructokinase and your glycogen phosphorylase are outside the mitochondria. Your lactate dehydrogenase H is inside the mitochondria, but its end product is pyruvate. So it converts lactate to pyruvate. Pyruvate dehydrogenase converts pyruvate to acetyl-CoA in the mitochondria. And that's illustrated in this diagram. So here you have pyruvate being produced through glycolysis. It can be taken up by the mitochondria, and through this pyruvate dehydrogenase enzyme, it can be converted to acetyl-CoA. So again, a review: once that acetyl-CoA gets formed in the mitochondria, it enters another metabolic pathway called the Krebs cycle, also called the tricarboxylic acid cycle or the citric acid cycle. It gets broken down. When you break down the acetyl-CoA, you get release of carbon dioxide. So understanding the Krebs cycle, you can understand where the carbon dioxide is produced that you exhale while you're exercising. So it comes from this pyruvate dehydrogenase reaction, and also from carbon dioxide produced in the Krebs cycle. So another useful thing with the Krebs cycle is it gives you an understanding of where energy comes from when you break down glucose, fats, or proteins. So you get one ATP that's produced directly in the Krebs cycle, but these molecules that are formed can also go to the electron transport chain and produce ATP. Now, another useful thing about the Krebs cycle, and I had this slide up last day, so I've mentioned these first three bullet points. The last thing that the Krebs cycle can tell you is how nutritional supplements might be involved in improving your aerobic metabolism. So specifically the B vitamins, riboflavin and niacin, and branched chain amino acids. So these molecules here are derived from those B vitamins, or their B vitamins are important for producing or helping to produce these molecules. And then with regards to branched chain amino acids, that comes from protein breakdown. Branched chain amino acids can enter different points of the Krebs cycle to help to refuel the intermediates of the Krebs cycle. So I'm going to cover that further in the protein metabolism section of the course. Now, this is the main part of the lecture, so this kind of helps you to review all the steps during the oxidation of one molecule of glucose. So the question here is, what is the total amount of ATP produced from the oxidation of glucose? And show where each ATP is produced. So we're going to go through all the little steps here involved in the oxidation of glucose and where the ATP is produced. So when you have glucose that enters from the blood into the muscle, it gets phosphorylated, gets converted to glucose 6-phosphate. That goes through glycolysis. With glycolysis, you get a net of 2 ATPs produced. So that's where the first ATPs come from. So you have glycolysis. This is in the cytoplasm. And you get 2 ATPs that are produced right there. Okay. So next, you'll get entry of pyruvate into the Krebs cycle. And notice there's 2 pyruvates. Pyruvate is a 3-carbon molecule. Glucose is a 6-carbon molecule. So you're preserving the number of carbons here. And then you go to acetyl-CoA, which is actually a 2-carbon molecule. So recall that you're losing your carbons because you produce 2 CO2s here. So this shows the Krebs cycle. Keep in mind that you have 2 pyruvates being converted to 2 acetyl-CoAs here. So for every molecule of glucose that's broken down, this Krebs cycle actually goes around twice. So first thing you'll notice is with the pyruvate dehydrogenase reaction, you have production of an NADH. And it actually is going to be 2 NADHs because you have 2 pyruvates here being converted to 2 acetyl-CoAs. So we'll add that to our total here. You've got the pyruvate dehydrogenase reaction. I'm just going to put P pyruvate DH reaction. And you get 2 NADHs. And recall when these NADHs get produced, they go to the electron transport chain. That's the next aerobic metabolic pathway. And you can see that you get 3 ATPs that are produced in this chain. So for each NADH that's produced, you get 3 ATPs. So I'm going to add the ATPs to that total. So in other words, we have 6 ATPs from that pyruvate dehydrogenase reaction. Next, we have 1, 2, 3 NADHs produced in the Krebs cycle. Recall the Krebs cycle goes around twice. And you also have 2 FADHs produced, 1 per Krebs cycle, but remember it goes around twice. So the FADHs, they produce 2 ATPs in the electron transport chain, as opposed to the NADH that produces 3. So we're going to add that to the total here. So you have a Krebs cycle. You're going to have 6 NADHs from that process times 3 ATPs in the electron transport chain equals 18 ATPs. And then you're going to have 2 FADHs times 2 ATP from the electron transport chain equals 4 additional ATPs. And then you'll notice from the Krebs cycle you get 1 ATP that's produced directly in the Krebs cycle. So again, the Krebs cycle goes around twice. So you're going to have 2 ATPs that are produced from that process. So within the Krebs cycle, you have 2 ATPs that are produced directly. Now, what we're missing here is 4 additional ATPs, and I'll quickly explain how those are produced. So within glycolysis, you have production of NADH, and that NADH can deliver its hydrogens to the mitochondria, but on the other side of the mitochondria, you have FADH that actually picks up the hydrogen and delivers that to the electron transport chain. So what you get here is you have a shuttle system involving NADH, FADH, that produces 2 additional ATPs to make 4 more ATPs, and that will give you your 36 ATPs in the whole process.